This is lecture 2.3, the second of our lectures on cell structure. You hopefully remember from our previous lecture that we looked at the structure of the cell membrane, how we could find it in any cell. And we also looked at the nucleus and the parts of the nucleus. So these 5 organelles of the cell you should already be able to identify. And now what we want to do is look at the cytoplasm of the cell. Now notice that cytoplasm is not one of the 12 organelles that you are to learn. Cytoplasm is a general term for everything inside the cell except the nucleus. So all of the other organelles and structures you see inside the cell membrane, except the nucleus, are collectively called the cytoplasm. I will not have you identify the cytoplasm in a diagram, but you may see the term cytoplasm in a test question. If I was to ask you what one of the organelles of the cytoplasm was, you would not name any of the parts of the nucleus. You would name the parts that we're about to investigate right now. So be sure you understand the term cytoplasm, but don't see it as something to identify in a diagram. Now, inside the cell membrane, outside the nucleus, are many, many organelles, lots and lots of structure. There are 7 structures, 7 organelles here that I would like you to recognize, and we're going to go through them one at a time. First, let's identify the mitochondrion. The mitochondrion is a very easily identifiable object. There are many mitochondrion in this picture. You see one pointed to right here, but there are a number nearby, and there are quite a few if you look throughout the picture. How do you identify a mitochondrion? Well, let's look at them up close, like this. One of the things that's so very obvious and different from everything else is their maze-like walls. The mitochondrion is actually made out of 2 membranes, one on the outside and one on the inside. If you look at any one of these mitochondria, you can see that distinction. And it turns out that the inside membrane is larger than the outside membrane, so it has to be folded in order to fit inside the smaller outside membrane. And those folds make it look like sort of a maze-like structure. I don't know if you remember going to a restaurant or having a little booklet of mazes you tried to get from the start to the finish, these little walls in here always remind me of that. The shape of the mitochondrion can be anywhere from round to sort of a long hot dog or sausage shape. And that's because, well, here's the full 3-dimensional look of the mitochondrion. You can see it cut away so that you can see the larger inside membrane. But notice, because it really is sort of a long hot dog shape, it depends on how it's cut as to what it will look like. So if you were to cut one of these across the length of it, you'd get a round sort of look to the mitochondrion. If you cut it lengthwise, you get sort of a long look. And if you cut it on an angle in some way, you'd have a third shape. But the shapes are always from sort of a sausage shape to a round shape. Just by way of a little bit of extra knowledge, we'll cover this in the lecture, the 4th lecture on cells, but this is where all of the energy within the cell is produced, or at least the majority of the energy that a cell utilizes for its activities are produced here in the mitochondrion. There are cells that have lots of mitochondria. There are cells that have a few. But every cell has mitochondria because every cell needs energy to run its various activities. Here are several of the cells that have quite a few mitochondria. If you look at the first cell picture there, the mitochondria, most of them are near the inferior part, the bottom part of that picture. If you look closely, you'll notice that the cell membrane folds in and out of that end of the cell, and the mitochondria and the cell membrane are all closely associated there. Makes a very complex end, but as you might guess, if these produce energy, there must be something at that end of the cell that requires lots and lots of energy for it to work. The center picture here, the muscle cell, of course, muscles utilize lots of energy in their movement, and you can see lots of mitochondria. In fact, they're so tightly packed in there that they have lots of funny shapes. They aren't so easily just sausage around. And the third picture there is sort of a little test. There are 2 mitochondria in that picture. The polymorphonuclear leukocyte actually has many mitochondria, but this slice through the cell doesn't show too many of those. But you should be able to find 2 in that picture. Look closely and see if you can find 2 objects in that picture that are mitochondria. Okay? And one of them is at about, I'd say 10 o'clock, if that was sort of a clock face. And one of them is way down around, say, 5 o'clock. They're small, they're round, but you can easily identify them. Nothing else in the cytoplasm has these maze-like walls of the mitochondria. Okay? So, we've identified mitochondria. Let's find a second organelle. This one is called a ribosome. And there are many, many ribosomes in the nucleus. Now, what am I pointing at there? Kind of hard to see. Let me take a expanded view of what we're looking at. Ribosomes are small dot-like structures found in the cytoplasm. Now, if you look closely at that picture I just put up, you can see some tiny, tiny, tiny little dots there. That's not what we're talking about. We're talking about separate, sizable black dots that you see here. Some of them are floating in the cytoplasm, but there are other places, like here, you can see them attached to the surface of sort of a couple of worm-like structures here. So ribosomes can be found in a number of places throughout the cytoplasm, either freely floating or on the surface of objects. Like right here, you can see it on the surface of that long sort of worm-like thing. Look closely at the nucleus. If you look at the surface of the nuclear envelope, you will see ribosomes attached there as well. Ribosomes are pretty much like the workers of the cell. They perform many of the complex production activities of the cell. And so there are jillions of these. They're all over the place on the surface of various membranes and free-floating throughout the cytoplasm. When you see ribosomes are very often, though, associated with these long worm-like structures that you see right here. Let's put this back in again, and let's talk about these worm-like structures. These long worm-like structures are called rough endoplasmic reticulum. I haven't spelled out endoplasmic reticulum in my picture here because there really isn't enough room to get everything in. You can see it spelled out in your yellow handout. On a test or a quiz, you must spell it out. I am not going to assume that you know what an endoplasmic reticulum is. So please spell it out. I'm just abbreviating here because I know what it is and I can do it. You have to prove to me that you know it. But what are they? Well, rough endoplasmic reticulum are these sort of channel-like, these little worm-like structures, sometimes divided, sometimes straight. These objects that you see there and they have little ribosomes dotted all over their surface. Okay? So you'll see them in many, many places. In fact, if you're looking at this picture, you see them all over the place. Many, many, many of these. And these happen to be the assembly lines of the cell. These are where anything that the cell is going to produce gets produced. These are like little factories in the cell. So there are rough endoplasmic reticulum all over the place, and you'll recognize them by their long, sort of skinny shape and the ribosomes attached to their surface. There is another type of endoplasmic reticulum that does not have ribosomes attached to the surface, and these are called smooth endoplasmic reticulum. In most cells, they are somewhat rare. There isn't a lot of them, and they tend to be smaller on the whole than the rough endoplasmic reticulum. But it's really the presence or the absence of the ribosomes that distinguish a rough endoplasmic reticulum from a smooth endoplasmic reticulum. In the cell picture that we're using here, there is very, very little smooth endoplasmic reticulum. One little bit of it way up here in this cell picture that we could identify. There are other cells that have quite a bit, but not that many. The smooth endoplasmic reticulum is not as obvious. Let's look a little bit more closely at these two and their relationship. You can see that rough endoplasmic reticulum is really more of not a little worm-like thing, but sort of a hollow pancake-like thing with chocolate chips over the surface. No, with ribosomes dotted over its surface. So this two-dimensional picture that we've been looking at like this is actually that three-dimensional picture that you see over there. Smooth endoplasmic reticulum, remember, is without those ribosomes on the surface. And actually this picture of the neuron, the nerve cell, has quite a bit of it in it. At this one end over here, I've just magnified it. Notice there are some very large pieces of smooth endoplasmic reticulum. They're even labeled. If you look at the letters SER, those are standing for smooth endoplasmic reticulum. But look up and down that little section right there, and you can see a number of very nice examples of smooth endoplasmic reticulum. There's also some over in this area, sort of over on the side, and this does present an interesting aspect of the two. You can see that very often smooth and rough endoplasmic reticulum are connected. If you look right below the letters SER, you can see rough endoplasmic reticulum in the picture. And if you gaze then to your left, follow that rough endoplasmic reticulum, you can see that when you get to one end there, it sort of runs out of ribosomes and sort of blends right into or begins to form then the smooth endoplasmic reticulum. So rough and smooth are related, and they are distinguished by the presence or absence of ribosomes on these long, sort of flat channel-like structures. So it's handy to study all three of these together. Make sure you can identify ribosomes, rough endoplasmic reticulum, and smooth endoplasmic reticulum. Good. So that's 4 out of 7, 3 more to go. Let's now look for what is called a Golgi apparatus. Some of you, when you were looking for smooth endoplasmic reticulum, might have mistaken this for that, because there is a very similar appearance to the two. The Golgi apparatus in this picture is right here. There's just one of them, and much of what is Golgi apparatus looks like smooth endoplasmic reticulum. The Golgi apparatus, too, is sometimes called a Golgi body or a Golgi complex. If you've taken a biology class, you may know it by any one of these names. The key word here is of course, Golgi. Let's look at it separately here for a moment. Here's an example of sort of a three-dimensional picture of what it is, sort of a stack of pancake-like objects, but when sliced through, they look like these long little slender channels, very, very much like the smooth endoplasmic reticulum. So that's the first thing you're looking at. What will typically distinguish this, though, is that there are usually 2, 3, 4 of these little flat tubes, or what look like tube-like structures, but they're all sort of tightly stacked together. True smooth endoplasmic reticulum will be separate sort of runs of that structure, not all tightly packed together like you see here. These tend to be short and very uniform too. You can see the edges of each one of these are all lined up pretty nicely. The other structure that you would see here are the little bubble-like structures that will always be found at the ends of the smooth endoplasm, or at the end of the Golgi apparatus. In fact, this Golgi apparatus is producing these little bubble-like structures that you see out at the ends there. So, Golgi apparatus has a distinct look to it. Doesn't look like any other of the organelles in the cytoplasm, although part of it might resemble smooth endoplasmic reticulum. But if you know what you're looking at, they're not easily confused. Here's a good little test for yourself. The neuron that I've put up here has 3 Golgi in the picture. Look closely at the neuron, and if it's not clear enough here in the lecture, pull out the picture of the neuron that you have from your handouts. See if you can identify the 3 Golgi apparatus in this picture. I'm just going to pause for a moment and let you look. Dum 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 dum. Now, if you need more time than that, pause me right now and then start us up again once you've found them. But let's find the 3. Okay, one of them is located right here. They're all fairly close to the nucleus here, but one of the Golgi in the picture is here. The second one is over here. And the third one is right there. So if you found those 3, you really, really know what you're looking for. That's good. You won't see a Golgi in every single cell picture that you have there, but some you can see more than one, like this one that we just looked at. Let me give you sort of a final exam now in Golgi. This is the polymorphonuclear leukocyte, and there is actually one Golgi apparatus in the picture. It's a little disorganized. It's not a perfect representation of what you would expect, but there is enough of it showing here that if you know what you're looking for, you should be able to pick it out. Okay? So, let me give you a moment to just look. Look. Look at the top, look over on one side, look over on the other side, down at the bottom, look somewhere in the middle. Pause me if you didn't have enough time, but go ahead and locate it right here. Notice it's not perfect, but if you look closely, you see something looking like short little stack, a little bit disorganized, but a short stack of what appears to be smooth endoplasmic reticulum, and you do see the little bubble-like structures there near either end. So that is the leukocyte, and that is its Golgi apparatus. All right, so that's now 5 out of 7. 2 last ones. Of all of the structures, the organelles in the cytoplasm, the 2 that sometimes get confused most often are these last 2. I've lectured through the 5 that can't be confused with anything else. If you don't know these, you don't know what you're looking for, because you can't confuse a ribosome with a mitochondrion or a Golgi with a rough endoplasmic reticulum. They just are what they are. But the last 2 on your list, lysosomes and vesicles, these 2 sometimes have the same appearance. Now, if you learn the lysosome first, because it just has one way that it looks, if you can find a lysosome, then anything and everything else that looks different is going to be a vesicle. So what does a lysosome look like? Well, there's one in this picture. I'm going to see if you can find it before I point it out. What you're looking for is a round object, small, not tiny, not like a ribosome or something, much larger than that, but a small round object. And when you look inside it, it's got a very grainy appearance to it. Lots and lots of tiny little dots in it. Very, very uniform. No big black spots in it or anything like that. Okay, pause me again if you need more time, but if you look around and around, look up, look down, side to side, and you should be able to find a lysosome right here. It's the one and only one in the picture. This is what a lysosome looks like. Let me blow it up a little bit. Not the 2 little objects with the black spots in them, but the one round object that has a nice, uniform, grainy structure to the center. That is the lysosome. Now, basically, every other small, round structure in this picture is a vesicle. So the vesicles are all of these guys, all of the ones up there near the lysosome. Vesicles are all over the place inside this alpha cell that you're looking at right now. So again, the easy way to do this is know that the lysosome has one and only one look to it. Vesicles can have a number of looks to them. They can look all black inside. They can look black and grainy. They can look white and grainy. They can look white and black. They can look all white. They can have any number of different appearances to them. But you want to be able to distinguish these 2 from one another. They can even have slightly different shapes. They might be a little oval or a little oblong, but they always have a certain amount of roundness to them. Let's look at some other vesicles, because again, vesicles are going to look very, very different from one another. If you look at the skeletal muscle cell here, at the bottom on the left side of the picture here, all those round black dots are vesicles. Vesicles are basically closets, garages. They're storage chambers. Something that the cell needs to collect and store and save and use are typically stored in vesicles. So like this muscle cell is storing some energy for itself. If you look at the other picture here, the adipose cell, obviously one great big giant vesicle, that great big black circle is a great big droplet of fat, and the job of an adipose cell is to store fat for you. So a vesicle is a name for an object within the cytoplasm that stores some kind of material. Let's look at one last cell that really is an excellent example of both lysosomes and vesicles. This, again, is the polymorphonuclear leukocyte. This leukocyte has within it many, many lysosomes. They are uniformly grainy, as we said before. They're not all perfectly round. This has within it vesicles. Remember, vesicles can have a variety of black and white sort of appearance to them. They can be all black, they might be all white, whatever. The picture that you're looking at, though, let me call your attention to something more than just lysosomes and vesicles. This is one of the most amazing pictures that you'll ever see. The cell pictures that you've been looking at here are actual photographs of real human cells. An artist did not draw this. Cells from some person's human body were frozen, they were sliced, and they were imaged by an electron microscope. Every object you see in this picture was exactly where it was when that real live cell was frozen. Now, what makes this picture so amazing is that this is a white blood cell doing what white blood cells do, frozen right in the midst of attacking, engulfing, and destroying bacteria. Now, look closely. Look at the top of the picture. You see those two round objects outside the cell membrane. Those are two round, what we call coccus bacteria, that are being attacked by this white blood cell. Notice how it is pushing its cell membrane out in sort of a gulping activity. Secondly, look over to your right of the picture, and you will see two more coccus bacteria that are in further stages of being engulfed. Notice how the cell membrane has almost completely enclosed around those two bacteria. And then look near the center of the picture and you will see two or even three other bacteria that are completely enclosed and engulfed and within a chamber inside the cytoplasm of the cell. Each of those round objects that has a bacteria in it would be called a vesicle. So the white blood cell attacks and destroys invading bacteria by literally engulfing them with its cell membrane. That cell membrane then forms a little chamber, and the bacteria is now trapped in the cytoplasm of the white blood cell. A further stage of activity here then is, if you look closely again inside, you will see the lysosomes, those round, light, grainy objects, are capable of fusing with those vesicles that hold bacteria. It turns out that that graininess inside a lysosome represents digestive enzymes that have been produced by the white blood cell. When the digestive enzyme, when the lysosome touches a vesicle, the two membranes unite and all of the digestive enzymes then mix with the vesicle and begin to literally take apart, digest, and destroy the bacteria. The end stage of this is when the bacteria is completely taken apart and is just a soup of organic chemicals, and the black circles that you see in the picture then are bacteria that have already been digested and are now fuel for the activities of this white blood cell. So the white blood cell protects us by destroying, digesting bacteria, and then uses those bacteria for its own energy purposes. An amazing, amazing cell, and an amazing, amazing picture of a white blood cell being frozen in the midst of its protective activities. As we've said before, let me just reiterate, structure determines function, doesn't it? All of these lysosomes and this ability to engulf these bacteria are an important part of what makes this cell do what it does. It could not do it without this structure. So, we've now identified 7 organelles that can be found within the cytoplasm of the cell. Make sure you know the 4 that belong to the nucleus, the cell membrane, and these 7 objects. Let me know, email me or whatever. Let me know if you have any questions. But other than that, this is how you identify in total the 12 organelles that I'd like you to know for this class. All right? Okay.